So I'd like to thank everybody for allowing me to come present some of our work today. As Brett mentioned at the very beginning, there's a number of us that he invited who really don't work directly in microbiota, and I am one of those guys. And I've been dragged kind of kicking and screaming into this field, but I will tell you a little bit about what we do in my lab, which is more stem cell biology and cell trafficking during inflammation. And I'll start by talking a little bit about its CD34 family and some new roles that it plays in inflammatory cell homing. And then I'll tell you about one particular model, an asthma model, where we can show it plays a really critical role in inflammatory disease. And then I'll tell you about a failed experiment where we were trying to show that CD34 plays a role perhaps in homing of inflammatory cells during neuroinflammation. And the only way we can reconcile this data is if there's actually a role for the microbiota in inducing this particular response. And then some general speculations on flora and inflammatory disease. So this is a family portrait of the molecules we study in my lab. CD34, as you heard, is a sialomucin, much like what you heard yesterday. It's a classic sialomucin with a lot of O-link glycosylation in the extracellular domain, a globular domain, stalk, and a short cytoplasmic tail with some consensus phosphorylation sites and a PDZ docking site at the end. Now, CD34 was discovered well over 25 years ago. Even today, it's still probably the best characterized marker of hematopoietic stem and progenitor cells, widely used in the clinic. If you've got lots of CD34 positive cells in a bone marrow transplant sample, you use it. If you don't, you don't, because you won't be able to get good transplantation of a patient. Now, despite that attention, despite the fact that there's over 20,000 papers citing CD34 as a stem cell and progenitor cell marker, really nothing is known about its function. And a lot of people have tried. In fact, TAC-MAC's group over 10 years ago knocked out CD34. Lasky's group in San Francisco's group, knocked it out as well, and found essentially no major phenotype. They were mind-numbingly boring. Right about that time, we cloned a second molecule in stem cells called potocolixin, realized immediately that it had very similar structure to CD34, and that if you knocked out this molecule, very likely this one could compensate. So we've been knocking out this molecule, as well as a third family member called endoglycan cloned in my lab, and slowly working up double and triple knockouts to really find out what these molecules are doing on stem cells. Now, this just shows you that all three of these molecules likely came from a common ancestral gene. They all have a similar genomic organization, even though they're on different chromosomes. And they all undergo a similar pattern of alternative splicing to give you molecules with either a full-length cytoplasmic tail or a truncated tail. And I'll come back to why that's important in just a couple of minutes. Now, what you'll read in the textbook about CD34 function is that it acts as a ligand for L-selectin and high endothelial venules. So textbook description, you've got high endothelial venules in the lymph nodes, which also express CD34. In the lymph nodes, they're endowed with a specific glycosylation that allows these molecules to be ligands for L-selectin. Naive cells roll along these L-selectin ligands, firmly adhere by integrins, and then diapedese into the underlying tissue. So that's the classic model of how these molecules work. That is certainly true for CD34 and potocolixin, but I want to point out that the only place in your body where you glycosylate these molecules for L-selectin binding is in the high endothelial venules in the lymph node, which is about 0.1% of all of the endothelial cells in your body. Stem cells do not glycosylate it for binding L-selectin, nor do most of the endothelial cells. So they must have a different function there. Okay, so trying to work up function from the knockouts. If you start looking, this is a complicated slide, but what I want to point out here is that there's some places where you have co-expression of these molecules and others where they have a unique distribution. The co-expression is on hematopoietic precursors and endothelial cells. And then you find a unique expression for each molecule. And in the case of CD34, we've recently shown that it's a beautiful marker of mast cells, a marker of eosinophils, and a marker of dendritic precursor cells, all molecules involved in inflammatory cell homing. So if you want to find CD34 function in a knockout, you should probably focus on these tissues where it's uniquely expressed and not some of these other places. So let's start in. This is CD34 expression on mast cells. It's pretty brightly expressed, in fact, not on the knockout mast cells. For the stem cell biologists in the audience, actually, these are two other stem cell markers. This is c-KIT and this is SCA1. These are all markers used to purify stem cells. Conversely, if you look at markers people use to purify away mature hematopoietic cells from stem cell preps like B220, MAC1, GR1, none of those are expressed by mast cells. So I think if you look at the literature and you find that hematopoietic stem cells have an amazing ability to repair damaged tissues and control inflammation, I think it's very likely that those effects in transplantation studies are due to contaminating mast cells in the prep. So beware of some of that literature. Now back to function. In terms of trying to find a function for CD34 in mast cells, we looked long and hard at the ability to differentiate, to proliferate, to degranulate, and the only major difference we could find in mast cells from 34 knockouts is shown here. Normally, mast cells like to grow as a nice single-cell suspension. If you take away CD34, you start to find cells that clump. And if you ask a graduate student to count several thousand of these cells, right before they quit, they will tell you that about 20% of the cells bind to each other, and there's about 5 to 10 cells per clump in this assay. That clumping is definitely CD34 dependent because if you put the gene back in using a retrovirus, you block adhesion. So in this case, it looks like CD34 is acting more like Teflon rather than Velcro. Now, there's only one other major sialomucin on mast cells, that's CD43. If you make a double knockout for both CD34 and CD43, now the effects become really obvious. The cells get incredibly adhesive, and once again, you can reverse that by expressing CD34 ectopically using a retrovirus. And the cytoplasmically truncated version blocks adhesion even better. So the model is that normally these molecules are expressed in a uniform shell on the surface of the cell. They tower above the integrins and other adhesion molecules and block nonspecific adhesion. If you take them away, now cells tend to aggregate or stick to other non-adhesive surfaces. Even in resting cells, in normal cells, if you activate them, you find that CD34 rapidly caps to one pole of the cell and exposes integrins for adhesion. So it's a movable Teflon. Now, we found that these molecules are expressed in inflammatory cells, eosinophils, mast cells, and dendritic cells. So we started looking in vivo for phenotypes during inflammation. And now, instead of having really boring phenotypes, we found that these molecules are really involved in a lot of different inflammatory cell processes. So for the CD34 knockout mice, we've looked at asthma, and we find that the mice are incredibly resistant to asthma. That's due to a failure of eosinophils and mast cells to traffic efficiently to the lung. As a Th2 disease, we've also looked at hypersensitivity pneumonitis, which is a Th1-based disease. There we find the mice are resistant to that as well. And that's due to a failure of dendritic cells to pick up antigen efficiently in the lung, migrate to the lymph nodes, and induce secondary responses. They're resistant to colon cancer and polyposis. That's due to a failure of mast cells to induce angiogenesis of nascent polyps. They're resistant to DSS-induced colitis. That's due to a failure of eosinophils to traffic to the gut. Salmonella infection as well. Now, they do still die from salmonella infection, and they do have the same number of bacteria when you infect them, but they don't have near the same amount of inflammatory disease as the wild-type mice. There is a downside to getting rid of CD34, so the mice are more sensitive to acute muscle damage due to a muscle satellite cell defect, and more sensitive to autoimmune and arthritis due to a vascular permeability defect, which I won't go into today unless anybody's really interested. So let me show you some really more detailed data on the asthma model, and then I want to come back to this multiple sclerosis model. Okay. So if you look at lungs at steady state in the 34 knockout mice, you find that they're actually quite similar to wild type. If you induce asthma using ovalbumin, by priming a couple of times and then waiting and do several intranasal instillations, wild-type mice get a huge increase in inflammatory cells in the lung. CD34 knockouts look pretty good. You can quantitate that by looking at bowel infiltration. So in naives, you have very few hematopoietic cells in the bowel, mainly macrophages. Induce inflammation via asthma, you get a huge increase, mainly in eosinophils. 34 knockouts are about a third as inflamed as wild-type. And again, the profile of cells, eosinophils, lymphocytes, and neutrophils, macrophages, is pretty similar in the 34 knockouts. The big decrease is in eosinophils. Mast cells are much rarer in lung, but they're potent, and they induce a lot of recruitment of other cell types. And similarly, you find a big increase in mast cells when you induce asthma, much fewer in the 34 knockouts. All of the defects I'm showing you here are hematopoietic dependent, so it's 34 expressed on hematopoietic cells, not on the vascular endothelia, and you can show that by giving a bone marrow transplant. If you transplant wild-type stem cells into a naive mouse, or CD34 knockout bone marrow into a naive mouse, the phenotype tracks with the hematopoietic cells, not with the background. You can induce airway resistance in mice, so when you get an asthma attack, you get constriction of the bronchi, and you can induce that in mice using a methacholine challenge. Wild-type mice, naives, and CD34 knockouts have a similar airway resistance. If you induce asthma in a wild-type mouse, you get a big increase in airway resistance. And the 34 knockouts are below baseline. So this second major hallmark of asthma is missing in the mice. Now, it's not because the mice can't make inflammatory cells. If you look in the bone marrow of allergic mice, you find the same frequency of B cells, progenitors, granulocytes, and macrophages, erythroid cells. Same number of eosinophil precursors, so it's not a failure to make the cells. If you start looking in the peripheral blood, though, it starts getting more interesting. So in wild-type naive mice, you have a similar number of eosinophils in the blood. That increases in allergic mice. It increases even further in the CD34 knockouts. So they actually make more and have more cells in the peripheral blood. But then when you look in the lung, in the parenchyma, you have less. And if you look in the airways, you have even less still. So it looks like the cells make it out, but then they have a failure to traffic into the lung tissue. If you take eosinophils out of the lung, the few that do make it there, and you compare them, for all the world, they look quite similar to the wild type. If you put them in a chemotaxis assay, though, then you notice some differences. So here's a transwell assay where you're asking eosinophils to migrate towards the ataxin. Wild-type eosinophils migrate just fine. The 34 knockouts are impaired. I think a lot of that is due to this Teflon effect and the cells being more sticky. We also have some recent data to suggest that maybe that's more of a specific defect also in chemokine receptor signaling and directional trafficking, which I could tell you about later if you're interested. Okay, one more piece of data on the asthma model. So we've taken the CD34 knockouts, crossed them to human CD34 transgenics that we got from Dan Tannen. Now these mice have the entire back locus for the human CD34 gene. And so now what we've generated is mice that lack mouse CD34 but have the human CD34. And it's expressed in all the right places, in the hematopoietic precursors, the vascular endothelia, and the inflammatory cells. And if you do that, wild-type mice get allergic, knockout mice are resistant, you recover the ability to develop asthma with the human transgenics. So the take-home message from this part of the talk is CD34 is required for inflammatory cell homing. In our hands, rather than acting like Velcro, it acts like Teflon. It's a blocker of nonspecific adhesion. We have some evidence that it's required for efficient chemotaxis in vitro, and as I mentioned, that looks like it might be a more selective effect on chemokine receptor signaling. Human CD34 can compensate for mouse CD34, and it seems to be important, particularly for mucosal inflammatory disease. Okay, now I'm going to switch gears to a model, the EAE model, which you heard a little bit about yesterday. This is a murine model for multiple sclerosis. It's a T-cell dependent autoimmune disease. You get T-cell, you inject the mice with a MOG peptide. It's a myelin protein peptide. You get attack of the myelin sheath and demyelination of some of the nerves. So you get short circuiting of the nerves, and the mice get paralyzed. You have CNS lesions, ascending paralysis beginning with the tail getting a bit limp, and then it progresses to hind limb and forelimb paralysis. This disease is well known to be modified by either stress as well as an environmental component, and people actually go to great lengths to co-house the mice whenever they do EAE because it's such a variable disease unless you do that. So that suggests there might be a commensal bacteria component as well. Okay, the model is you inject with MOG peptide with pertussis toxin on day 0. You boost with pertussis toxin 2 days later, and then you wait for the next 3 weeks and start monitoring the mice for signs of paralysis. Stage 1 is tail weakness, hind limb weakness. 3 is partial paralysis in the hind limbs. 4 is complete hind limb paralysis. 5, the mice get completely moribund and you have to euthanize them. Now, the reason we were interested in this model, as I mentioned, we were interested in mast cell-dependent diseases, and a paper came out quite some time ago suggesting that mast cells are essential for EAE. This is a paper in JXMed, and there's been several follow-up papers in JI and European JI showing some of the details of why the mice are resistant if they lack mast cells. And here's some of the data from that paper. So if you take mast cell deficient mice, these are WWV mice which have a mutation in c-KIT which renders them completely deficient in mast cells, and you compare them to wild-type mice and you induce EAE, you get a much higher clinical score with wild-type mice. The mast cell deficient mice are protected. That's in terms of the total mice. You also find a reduced incidence of disease in the mice you try to give EAE to. And the beauty of this model is actually you can take the WV mice and transplant in bone marrow-derived mast cells and now reconstitute the mice and look at how that changes the disease. And what these researchers showed was that the wild-type, the mast cell deficient mice reconstituted with mast cells now recover the sensitivity to the disease. So it looks like a nice, neat story. We thought this was going to be a slam dunk. We just take our mice, we put them in the model, and we show that CD34 is required for trafficking during EAE. So this is our first experiment where we tried to compare the wild type and knockout mice, and we got no effect. And in fact, the 34 knockouts, we've done this 5 times, they either were identical to wild type or they got slightly worse disease compared to wild type. So I pushed the postdoc, Jamie Bennett, who's fantastic at doing this EAE model, to go back and do it again, this time reconstituting the mice with either wild type or 34 knockout mast cells. And again, using WSASH, this is a different KIT mutation on a BLAK6 background, exactly same disease whether you've got mast cells or not. Here's the WV mice, which are on a different background, but again, exactly the same disease. And in fact, it looks like the WV mice without mast cells did even worse than the mice with mast cells. So we can't find an effect of mast cells. Again, we've repeated this, just comparing BLAK6 to SASH. WV did their litter mates, no big defects. And we even went to the length of actually asking a friend of ours in a different lab, Paul Koobs, who had been working in a similar model, to look in their colony and see if they could induce EAE, and they couldn't either. So 2 different labs, completely different sets of reagents. Neither of us can reproduce this model. Okay, so that's kind of where that story stopped for a while, and we thought, you know, the only way we could possibly explain those differences is either there's a difference in the biota in the mice. Maybe there's something that's making the mice in somebody's colony more stressed, or they have a different flora. Most of the model I showed you that showed a defect in EAE without mast cells was done in a conventional facility. Then out came these papers, which you've heard extensively in this colloquium, showing the Th17 cells play an important role and are induced by segmented filamentous bacteria, and a more recent paper showing that segmented filamentous bacteria can induce Th17 cells, which then are important for EAE. These were using germ-free mice largely, and it showed that SFB colonization induces susceptibility. So we thought, okay, maybe mast cells are playing a role in regulating SFBs. So we had a quick look at our colony and PCR'd them for SFBs, and we found, quite interestingly, that all of the mice in our BLAK6 colony, and these have been maintained for 3 years in our colony and crossbred periodically to make sure they're on a good background. All of our BLAK6 colonies had SFBs, and 7 out of 10 boxes of WSASH mice that we analyzed lacked SFBs. So that started to sound pretty interesting. And it's been reported that if you get mice straight from JAKs from their particularly clean unit, they lack SFBs, and we verified that as well. So we tried another experiment, and again, this was kind of a crude shot at this, but we received some mice from JAKs, either WV or BLAK6, confirmed that they lacked SFBs on arrival, waited a week, made sure they were still SFB negative. Then we co-housed them with some of our own BLAK6 mice, which we knew had SFBs, showed a week later that they had acquired them. and we also maintain a stock that lacked SFBs. Orally gavage the mice with a poo shake from some of the mice that had SFBs to make sure they got well colonized, confirmed that they had SFBs, and then induced EAE and monitored disease course. And unfortunately, as I mentioned, this is negative data. We, again, still could not see a difference between mice that do or do not have mast cells, whether they had SFBs or not, or whether they had mast cells or not. No difference in disease course. Now, there could be a lot of reasons for that. One is that maybe the adult response is already fixed, and maybe we really need to colonize the neonates or cross-nurse the mice in order to make sure that they've gotten the SFBs very early. Fetal exposure seems like it may be may give us more lasting effects that we can now see in the mast cell deficient mice. Interestingly, there are examples where even maternal exposure or perinatal exposure to bacteria can have a very dramatic effect on skewing immune response. In particular, in the allergy field, there's some new data that suggests if you immunize pregnant female mice with bacteria, you can actually skew the immune response and protect the offspring from development of asthma. So maybe we need to look earlier. Perhaps we need cleaner mice. So initially the effects of SFBs have been observed in germ-free mice and clean mice. Maybe we need to make our mice germ-free, or maybe we need to make our mice a little bit more dirty. And as I mentioned, the first place where people have shown mast cells were important for EAE was in non-SPF mice, conventional mice. And lastly, as Bill mentioned yesterday, maybe we need to have a good close look at the microbiota in our strains, not just looking for SFBs, but really looking at the total microbiota using TRF-LPs or other techniques to really scan. The good thing about having worked on a lot of inflammatory disease models and having to prove whether mast cells or eosinophils or other cell types are involved in inflammatory diseases, we've got a great collection of mice to start surveying for microbiota. 34 knockouts I've mentioned. We have all of these mice, which include molecules involved in trafficking, enzymes involved in inflammatory disease and signaling molecules, chromatin remodeling genes, mice that lack mast cells, mice that have hyperactive mast cells, eosinophil deficient mice, platelet defective mice. And I think if we start scanning these mice and looking deeply at the microbiota they're colonized with, then we might start getting some clues as to whether this chicken or egg phenomenon, which is more important, the genes or the microbiota, and which is leading which. Thanks to the mice, so that's one strain of mice that we have in our lab. We don't know what this mutation is, but it makes the mice pretty cute. These are our bigger friends. These are the people we've been collaborating with. I've mentioned the people in my lab. Jamie Bennett does the EAE model, fantastic at doing that model. Marie-Renee Blanchet did all the asthma data with help from Matt Gold in my lab, all the asthma data I showed you today. Mike Hughes has picked up the microbiota story and some of the asthma and some of the EAE data. Collaborators in Vancouver and elsewhere, and our funding agencies. And I will stop there.